Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2016. If you are new to my channel, please do like, share and subscribe. Then coming to the year June 2016. E. coli is being grown in a medium containing both glucose and lactose. On depletion of glucose, expression of beta-galactosidase it will increase because that is the only source of carbohydrate which is present in the medium for utilization by E. coli. So answer is option 2. Then error-free repair of double-strand breaks in DNA is accomplished by homologous recombination. Answer is option. Then next one. Histone deacylase catalyzes the removal of acetyl group from the N-terminal of histones. Which amino acid of histone is involved in this process? It is lysine. Answer is option 1. The next one. RNA interference is mediated by both siRNA and miRNA. Which one of the following statements about them is not true? Both siRNA and miRNA are processed by dicer. That is correct. Both siRNA and miRNA usually guide silencing of the same genetic loci from which they originate. That is not correct. miRNA is a natural molecule while siRNA is either natural or a synthetic one. That is also correct. miRNA but not siRNA is processed by DROSHA. That is also correct. So the option here is, answer here is option 2. Then next one, that is part C questions. E. coli was grown in 3 different experimental conditions. In one, it was grown in medium containing glucose as carbon source. In the second, in medium containing both glucose and galactose. In the third, was infected with phage. Match the curves shown below with the treatment. So the C curve should be the one which is infected with phage. That is why there was a growth there, then there was a decline in the number of cells. So C must be infected with phage. Then B should be the normal growth curve. That is in the case of glucose. And A will be is having 2 exponential phases. So it must be galactose and glucose. So the answer here is option 2. A is grown in glucose and galactose. B in glucose. C is infected with phage. Answer is option 2. Then next one. Each amino acyl tRNA synthetase is precisely able to match an amino acid with the tRNA containing the correct corresponding anticodon. Most organisms have 20 different tRNA synthetases. However, some bacteria lack the synthetase for charging the tRNA for glutamine, which is with its cognate amino acid. How do these bacteria manage to incorporate glutamine in their proteins? So the answer is from option 3. In these bacteria, the amino acyl tRNA synthetase specific for tRNA glutamic acid also charges tRNA glutamine with glutamate. A second enzyme then converts the glutamate of the charged tRNA glutamine to glutamine. So the answer here is option 3. Then next one. As topoisomerases play an important role during replication, a large number of anti-cancer drugs have been developed that inhibit the activity of these enzymes. Which of the following statement is not true about topoisomerases as a potential anti-cancer drug target? Now topoisomerases, they remove supercoiling of the DNA. As cancer cells are rapidly growing cells, they usually contain higher level of topoisomerases. That is correct. They have asked for the statement which is not true. The transient DNA breaks created by topoisomerases are usually converted to permanent breaks in the genome in the presence of topoisomerase targeted drugs. That is also correct. As cancer cells often have impaired DNA repair pathways, they are more susceptible towards topoisomerase targeted drugs. That is also correct. The drugs which specifically target topoisomerases usually do not affect normal fast growing cells. They will target both cancerous and normal cells. So option 4 is incorrect. Then next one. Some errors occur during DNA replication that are not corrected by proofreading activity of DNA polymerase. These are corrected by specialized repair pathways. Defects in the activities of some of the following enzymes impair this process. So they have asked about mismatch repair enzymes have been asked. So DNA polymerase 3 and DNA ligase that is involved in mismatch repair. MUTS and MUTL, they are also involved in mismatch repair. So the answer here is option 4. Both A and C is the correct answer. Then next one. An eukaryotic cell undergoing mRNA synthesis and processing was incubated with P32 labeled ATP with the label at the beta position. Where do you think the radioactive isotope will appear in the mature mRNA? Now P32 will not appear in the mature mRNA. It will appear because the first nucleotide at the 5' end contains 3 phosphate groups and the rest has only 1 phosphate group. So it will appear in the mature mRNA, but the condition should be that the P32 labeled ATP is used. So the first base should be adenosine at the 5' end. Then it will appear. So your option, that is option 3, that is P32 will appear at the 5' end of mRNA if only it has A as the first nucleotide. Then next one. One of the cellular events that TOR kinase positively regulates is the rate of rRNA synthesis. TOR regulates the association of a transcription factor to a Pol1 subunit. So what does TOR do? It regulates the association of a transcription factor to the Pol1 subunit. When the TOR is inhibited by drug rapamycin, the transcription factor dissociates from Pol1. A yeast strain is engineered which expresses a fusion of transcription factor and the Pol1 subunit. So it is a fusion construct that they have made. The level of rRNA synthesis is monitored in these cells during pulse labeling following rapamycin addition for the times indicated below. So if you add rapamycin, the TOR is inhibited. How? The transcription factor dissociates from the Pol1. The transcript profile of the rRNA observed for the wild type cell is given below. This is for the wild type cell. Identify the pattern expected in the engineered strain. Now in the engineered strain, one fusion of the transcription factor and the Pol1 subunit is expressed. So you add the drug or do not add the drug, the transcript profile remains the same. Whereas in the wild type, it decreases with time. That is why here in the wild type, it is decreasing with the time. Whereas in option 2, if you see, it is showing the same. Whether you add the drug or not add the drug, it remains the same because it is a fusion construct. Then next one. Which of the following are not transcribed by RNA polymerase 2? So tRNA and 5S rRNA are not transcribed by RNA polymerase 2. They are by RNA polymerase 3. So answer is option 4. Then next one. RNA editing, a post-transcriptional process, is achieved with the help of guide RNA. Which one of the following statements about the process is not true? So guide RNA is dependent RNA editing happens in the kinetoplast DNA. Kinetoplast DNA is your mitochondrial DNA, so it is true. Guide RNA is involved in chemical modification of tRNA. It is not tRNA, it is mRNA. So, which is not true is option 2 is the right answer. Then next one. Telomerase, a RNA protein complex which completes the replication of telomeres during DNA synthesis, is a specialized. It is a specialized reverse transcriptase. So it is RNA dependent DNA polymerase. Answer is option 1. Then next one, that is part C questions. Addition of antibiotic cephalexin to growing E. coli cells lead to filamentation of the cells followed by lysis. Cephalexin is an inhibitor of peptidoglycan synthesis. Answer is option 3. Then next one. Fluorescently tagged protein was used to study protein secretion in yeast. Fluorescence was observed in Golgi, secretory vesicles and rough endoplasmic reticulum. Which of the following describes best the sequence in these events? So, from the rough endoplasmic reticulum, it will be to the Golgi, and from Golgi to the secretory vesicle. So, CAB. Answer is option 3. Then next one. In order to ensure that only fully processed mature mRNAs are allowed to be exported to cytosol, pre-mRNAs associated with snRNPs are retained in the nucleus. So snRNPs, it helps in splicing. So those are retained, pre-mRNAs associated with snRNPs are retained in the nucleus. To demonstrate this, an experiment was performed where a gene coding a pre-mRNA with a single intron was mutated either at the 5' or 3' splice site or both the splice site. Given below are 4 possible outcomes. Choose the correct combination of the possible outcomes. So first one, pre-mRNA having mutation at both the splice sites will be retained in the nucleus because of the presence of bound snRNPs. Because of mutation at both the splice site, snRNP will not be able to bind. So option A is not correct. Pre-mRNA having mutation at both the splice site will be exported to cytosol because of the absence of bound snRNPs. That is correct. Pre-mRNAs mutated at either 5' or 3' splice site will be retained in the nucleus. That is correct. Because it is either 3' or 5' splice site. Because of the presence of bound snRNPs. snRNP has got one site free for it to bind whether the other site is mutated. If the other site is mutated. So C is correct. So the answer here is option 1, that is B and C. Then next one. Polysome profiling of cells treated with 3 hypothetical translation inhibitors is shown in the plot below. These 3 inhibitors are CHP, leaky inhibitor of translation, LTM, arrest ribosome at the initiation codon, PTM, inhibit ribosome scanning. Match the polysome profile to the inhibitor. So 1 will be A. Why? Because leaky means some protein is synthesized. So polysome will be formed. So this must be 1. 1 must be A. Then LTM, it arrests ribosome at the initiation codon. So till 8S will be formed, but polysome will not be formed. So B2 or 2B. Then PTM, inhibit ribosome scanning. 8S will not be formed as scanning is inhibited. So the answer here is 1A, 2B and 3C. Then next one. In mammals, CG-rich sequences are usually methylated at C, which is the way for marking genes for silencing. Although the promoters of housekeeping gene are often associated with CpG islands, yet they are expressed in mammals. Which one of the following best explains it? So housekeeping genes, they are expressed in all the cells. So unlike within the coding region of a gene, CG-rich sequences present in the promoters of active genes are usually not methylated. So answer is option 3. Then next one. Telomerase, a protein complex, has a special reverse transcriptase activity that completes replication of telomeres during DNA synthesis. Although it has many properties similar to DNA polymerase, some of them are also different. Which one of the following properties of telomerase is different from that of DNA polymerase? Sorry. Telomerase requires a template to direct the addition of nucleotides. Telomerase uses RNA template. And telomerase can only extend 3' OH end of the DNA. Telomerase does not carry out lagging strand synthesis. Yes, DNA polymerase synthesizes both leading and lagging strand, but telomerase carries out only leading strand synthesis. So 3 is correct. Answer is option 3. Now next one. In eukaryotes, a specific cyclin-dependent kinase activity is required for the activation of loaded helicases to initiate replication. On the contrary, this CDK activity inhibits the loading of helicases onto the origin of replication. Considering the fact that during each cycle there is only one opportunity for helicases to be loaded onto origins and only one opportunity for these loaded helicases to be activated, which one of the following graphs best depicts the CDK activity in G1S phase of the cell cycle? Now pre-initiation complex, it forms at the origin of replication and it takes place in the G1 phase. So CDK activity is low in the G1 phase, whereas pre-replication complex activates during S phase, and it is at this phase that CDK activates the pre-replication complex, which means CDK is more in the S phase. Or I can say pre-replicative complex forms in the G1, and when it moves from G1 to S phase, CDK gets activated. So there is no CDK activity in G1 phase, whereas CDK activity is more in the S phase. So the answer here is option 1. Then next one. In trympanosomes, a 35 base pair leader sequence is joined with several different transcripts making a functional mRNA. The leader sequence is joined with other RNAs by the process of trans-splicing. And trans-splicing is a form of RNA processing where exons from two different primary RNA transcripts are joined end to end and ligated. So the answer here is option 2.